Hi dear students, in this video I am going to explain C2 cycle or photorespiration, which is an unusual process which is taking place in photosynthetic tissues. What is photorespiration? It is an unusual metabolic pathway responsible for photosynthetic oxygen uptake and the light dependent release of carbon dioxide. So it is termed as photorespiration. Normally in photosynthesis, carbon dioxide is taken in and oxygen is released. But here in this particular process, oxygen uptake and carbon dioxide release takes place. So because of this gaseous exchange which resemble respiration, it is termed as photorespiration. This happens in C3 plants during high temperature and high oxygen concentration than carbon dioxide. That means low carbon dioxide oxygen ratio. The reason for photorespiration is the unusual property or dual property of Rubisco to catalyze oxygenation of RUBP sometimes instead of carboxylation of RUBP. Rubisco catalyze oxygenation of RUBP under high temperature and high oxygen concentration in cell. That means low carbon dioxide oxygen ratio. It's a waste process or unwanted process that is added to Calvin cycle in some situations, which reduces the efficiency of the carbon fixation of Calvin cycle. Krotko in 1963 coined the term photorespiration to explain the light-dependent carbon dioxide release in photosynthetic tissues. The first stable compound in this cycle or photorespiration is phosphoglycolate, which is a 2-carbon compound, so that this photorespiration is also known as C2 cycle. Since this cycle involves metabolism of glycolic acid or glycolate, this pathway is also known as glycolate pathway. Where C2 cycle operates? Obviously, this cycle starts in chloroplast because the first phase of C2 cycle is oxygenation of RUBP. Since RUBP and Rubisco are present in the stroma of chloroplast, this cycle starts in chloroplast, but later it proceeds into two different cell organelles also, that is peroxisomes and mitochondria. So, in order to complete C2 cycle, three different cell organelles are needed: chloroplast, peroxisome and mitochondria. Let's learn about the dual nature of Rubisco. I hope all of you remember about Rubisco, what we have learned about Rubisco in Calvin cycle. Rubisco is ribulose 1,5-bisphosphate carboxylase bar oxygenase. It is an unusual enzyme which possesses a dual nature. It possesses the ability of both carboxylation and oxygenation of RUBP. Under high cellular carbon dioxide concentration and in normal temperature, Rubisco acts as carboxylase or Rubisco catalyze carboxylation of RUBP. So when Rubisco catalyze the carboxylation of RUBP, the active site of Rubisco is occupied by carbon dioxide. So carboxylation takes place. So when carboxylation takes place, this 5-carbon compound combines with this 1-carbon compound, and these 6-carbon addition compound splits into two 3-carbon compounds, 3-PGA, two molecules of 3-PGA. So the normal C3 cycle operates in stroma of chloroplast. But in some situations, that means under high cellular oxygen concentrations, high temperature, etc., Rubisco shows a changed affinity, that is towards oxygen. So in such situations, oxygen shows more affinity towards Rubisco, or Rubisco shows more affinity towards oxygen. So oxygen occupies the active site of Rubisco and oxygenation of RUBP takes place. As a result of oxygenation, here no carbon is there. The 5 carbon here, it splits up into one 3 carbon compound, that is the same one here, 3 PGA, and in addition to one molecule of 3 PGA, here a 2 carbon compound is forming, that is 2 phosphoglycolate. This particular compound 2 phosphoglycolate, it acts as the starting compound of a new cycle, which is C2 cycle. So this process occurs because of the oxygenation function of Rubisco. This happens when high cellular oxygen concentration and high temperature present. So this dual nature of Rubisco is the reason for photorespiration or C2 cycle. This is the simplest representation of photorespiration. Here, from this representation, it is clear the C2 cycle operates in 3 different cell organelles: chloroplast, peroxisome and mitochondrion. Here the reaction starts in chloroplast because Rubisco is here. The substrate of Rubisco is RUBP. So in the conditions of under high temperature and high oxygen concentration, more affinity is showing towards oxygen by Rubisco. So oxygenation of RUBP takes place. So RUBP combines with oxygen. So only 5 carbon is present. So these 5 carbon splits into one 3 carbon compound and one 2 carbon compound. The 3 carbon compound is the same compound that is produced in Calvin cycle, that is the 3 PGA, 3 phosphoglycerate, and the other compound is 2 phosphoglycolate. This is the starting compound of C2 cycle. So here the 3 PGA formed in the chloroplast, or it is in the stroma. So it is added to the 3 PGA pool of Calvin cycle. So normal Calvin cycle is operating here. But here 2 phosphoglycolate formed here. It starts a new cycle. So a new cycle is starting from this 2 phosphoglycolate, and the newly starting cycle from this 2 phosphoglycolate, it is termed as photorespiration or C2 cycle. So from this picture it is clear C2 cycle is not a separate cycle from C3 cycle, but it is an added cycle to C3 cycle. So C3 cycle is also operating here, but it is sidelined. So C2 cycle is also operating, or it is also added to C3 cycle. So because of this process, the efficiency of C3 cycle is reducing. Okay, so here the exact process I will explain later. So the reaction proceeds to peroxisome, then to mitochondria, and then it returns back to chloroplast. So the most important point is here in mitochondrion, one important process is taking place, that is the release of carbon dioxide and also ammonia. So this release of carbon dioxide, this is the most significant process happens here. Because of this point, or because of this process, we consider photorespiration as a waste process, because in chloroplast carbon dioxide is the most precious molecule which is to be fixed as glucose through dark reaction. But here, through this process of photorespiration, here the most important or precious carbon dioxide molecule is released as a waste material, where in mitochondrion. So what is the significance of mitochondrion? Mitochondrion is the site of respiration. In the context of respiration, carbon dioxide is not a precious molecule. It is a waste process. It is a waste product. So here, as a result of photorespiratory pathway, the most precious carbon dioxide is lost in mitochondrion as a waste product. Because of this process, because of this release of carbon dioxide in mitochondrion, photorespiration is considered to be a waste process or unwanted process which reduces the photosynthetic efficiency of C3 plants. Okay. So this is a very simple representation. Now let's learn the actual process in detail. So this is the actual representation of C2 cycle, where we can see three different compartments: chloroplast, peroxisome and mitochondrion. Here the reaction starts here. RUBP is there. So we know this process takes place in high temperature and high oxygen concentration. So in such situation, Rubisco acts as oxygenase. So RUBP, oxygenation takes place. RUBP combines with oxygen. So as a result of this oxygenation, instead of 3 PGA molecule, here 1 PGA, that is a 3 carbon compound, and 1 2 carbon compound phosphoglycolate is forming. This acts as the starting compound for the C2 cycle. That is why the name C2 cycle, because of this 2 carbon compound. So what happens to this phosphoglycolate? What happens to this 3 PGA? 3 PGA normally it is present in the chloroplast, so enter into Calvin cycle. So the phosphoglycolate formed here, it enter into the C2 cycle pathway or photorespiratory pathway. So what happens to phosphoglycolate? The first process is a dephosphorylation process. That means a removal of phosphate group by the enzyme phosphatase. Phosphatase enzyme removes this phosphate group from here. So glycolate is forming by the removal of phosphate group. Now glycolate is formed. No difference in the carbon amount. So here also 2 carbon compound. Okay. Now the glycolate formed in chloroplast leaves chloroplast and migrates into peroxisome. So glycolate now migrates into peroxisome. So in peroxisome, glycolate undergoes oxidation process. Oxidation process. Glycolate undergoes oxidation process in peroxisome. So as a result of oxidation, glyoxylate is forming. No difference in the carbon content. So again 2 carbon. So glyoxylate is now forming. Along with this glyoxylate, H2O2 is also forming here. As a result of this oxidation, H2O2 is also releasing. So from this point it is clear why this reaction is taking place in peroxisome. Because in this particular process, H2O2 is released as a side product. Here H2O2 is a highly toxic material which is soon removed in peroxisome by the activity of an enzyme catalase. Catalase enzyme is plentifully available in peroxisome, so which helps in the removal of this H2O2. By the activity of catalase, H2O2 soon converted to H2O and half O2. Okay, so this glyoxylate now present in peroxisome. Next, it undergoes a transamination process. Transamination processes are very important in metabolism. They are a particular type of reaction in which an organic acid reacts with an amino acid and the amino group from the amino acid is transferred to this organic acid and the COOH group from the organic acid is transferred to amino acid. So because of this amino group interchange or carboxyl group interchange, a new amino acid and new organic acid is forming. Such reactions are transamination reaction. So here glyoxylate is a 2 carbon organic acid. So the functional group here is COOH. Glutamate is a 5 carbon amino acid. So here amino group and COOH groups are there. So here what happens? This is a transamination process. So transamination, transfer of amino group. So amino group from glutamate is now transferred to this glyoxylate and COOH group from glyoxylate is transferred to glutamate. So what happens? Now this 2 carbon organic acid is now converted to a 2 carbon amino acid and the 5 carbon amino acid is now transferred to a 5-carbon organic acid. Okay, so the 2-carbon amino acid now formed is glycine. This is the simplest amino acid, glycine. So glycine is formed here, and the newly formed glycine now it leaves peroxisome and enter into mitochondrion. Okay, so now one glycine molecule reached mitochondrion. Now it waits for the next glycine to reach there. So when two molecules of glycine reach there, two molecules of glycine interact with each other. So two molecules of glycine means now 4 carbon is present. So these two molecules of glycine interact with each other, and out of these 4 carbon, 1 carbon is released as carbon dioxide, and the remaining 3 carbon rearranges to form a new amino acid called serine. So a 3-carbon amino acid serine is forming, and the remaining 1 carbon is released as a waste product in mitochondrion, which is released outside as a waste material. And here we know 2 amino acid molecules are there, 2 glycine. Okay, so 2 amino groups are there, but here only serine is another amino acid, 1 molecule of serine, so 1 amino group is needed. So 1 amino group is here become free, and that amino group is released as ammonia. Okay, so here serine is formed. Now it is present in the mitochondria. So serine formed in mitochondria now leaves mitochondria and migrates to peroxisome again, and peroxisome. So serine now reached peroxisome, which is a 3-carbon compound. Now next phase is again transamination. So this is an amino acid. It reacts with an organic acid, alpha ketoglutaric acid. So what happens? Amino group from here, it is transferred to this 5-carbon organic acid, and COOH group from here it is transferred to serine. So the resultant products are a 3-carbon acid, that is hydroxypyruvate, and a 5-carbon amino acid, glutamate. Okay. Now the 3-carbon acid hydroxypyruvate in peroxisome, it undergoes a reduction process or hydrogenation process. That means addition of hydrogen. So by the addition of hydrogen to hydroxypyruvate, glycerate is forming. So for the addition of hydrogen, coenzymes are needed. In peroxisome, the available coenzyme is NADH, not NADPH, it is NADH. So one molecule of NADH is utilized here, and hydrogen is donated. So using this hydrogen donated by NADH, glycerate is forming, which is a 3-carbon acid. So the newly formed glycerate, it is now leaves peroxisome and enter into chloroplast. So inside chloroplast now glycerate is present. So this glycerate next it undergoes a phosphorylation process with the help of ATP. When an ATP breaks or undergoes hydrolysis, and the last phosphate group is removed and it is added to this glycerate. So it forms PGA, phosphoglycerate. Glycerate now forms phosphoglycerate, PGA, by the hydrolysis of ATP. So 3-PGA, 3-carbon compound, it is formed. So now this 3-PGA is present inside the stroma of chloroplast. So naturally it enter into the Calvin cycle pathway. So we reached again chloroplast. So the cycle is now ended. So this is the C2 cycle. So here, why this photorespiration or why this cycle is considered as a waste material? Only because of this release of carbon dioxide here. So here actually carbon is a very precious material or resource for the glucose synthesis. So this precious carbon is here lost as a waste material in mitochondrion. And here how much carbon is lost. Out of the 4 carbon that is reaching mitochondrion, only 1 carbon is lost as waste material. The remaining 3 carbon reaching or returning back to chloroplast. So that is because we can say 25% of carbon dioxide is 25% of carbon is lost as carbon dioxide. 75% of carbon is returning back to chloroplast. Okay, so this is the cycle of photorespiration or C2 cycle. What are the disadvantages of photorespiration? Photorespiration is an unwanted waste process which operates in C3 plants, which interfere with the Calvin cycle and adversely affect photosynthetic productivity. That means reduces photosynthetic efficiency by 30 to 40%. It results in wastage of energy by consuming ATP and NADH and carbon as carbon dioxide in mitochondria without producing energy-rich compounds. According to Hatch and Slack, photorespiration is a metabolic adjunct to Calvin cycle. That means it has been added to Calvin cycle, but essentially it is not its part. So it is added to Calvin cycle. So because of this addition, the photosynthetic efficiency is much reduced. Is there any advantage for photorespiration? Not that much advantages are known for photorespiration. The only suggested advantages, it brings about the production of amino acids, glycine and serine. I hope all of you understood about photorespiration well. Thank you.